Our second scientist is Aditi Falnikar. She will be presenting work from Dr. Richard Vallee's lab here at Columbia University. Dr. Vallee has been studying various aspects of the KIF1A gene and kindly presented at our first ever family conference 2 years ago, where he provided us with our first glimmer of hope then, discussing how BDNF had a positive effect on fetal rat brain tissue. I'm thrilled to hear about the progress since then. Please welcome Dr. Vallee and Aditi Falnikar.
Thanks. It's a pleasure to be here again. That was Dominique. That was one of the best talks that I've ever heard on motor proteins. You have a talent, and your research as well. Is this on?
Hold that. It's hard to hear you. Can you hold that closer?
Yes. Did you hear kind of what I said? Yeah. Thank you, Dominique. Beautiful talk. Originally I was going to give this talk. That's why I'm here. I'll just take a minute. I just want to give a little history of how we became involved in this overall project. My lab has worked on the motor proteins for many years, and about 2 or 3 years ago, I received an email. We had published a paper on KIF1A in the brain, and I received an email from a person, a woman in the St. Louis area, and she said that her grandchild had a mutation in KIF1A. And we corresponded a little bit, but ultimately this led subsequently to, I guess, Luke contacting me again and saying, at this very institution where I was working, there's going to be a meeting of parents and a discussion of children. And so I attended that, and and we became increasingly involved. Okay. We have been working on KIF1A for several years, and we discovered a couple of important functions of KIF1A, especially in the rat and mouse brain, but also presumably in humans. And to answer one of Dominique's questions that she posed, there are indeed many known cargoes for KIF1A, and one of them is it's a hormone or a growth factor, referred to as brain-derived growth factor. You don't have to memorize that. Aditi will be talking about it extensively. And our research, her research, has indicated that BDNF is a particularly important factor perhaps in disease. So Aditi is a senior research associate in my lab, and she is the main person, as you heard from Dominique. We do not have factories in medical schools. Each student, each postdoctoral person, each research associate will tend to become an expert, even the world's expert in the particular problem they're working on. So Aditi will show you some of what she's been doing and give you the real interesting stuff that I'll skip. Okay.
Good afternoon, everybody. I'm honored to be here. Thank you for giving me this opportunity to share my research findings with all of you. So today I'm going to talk about evaluation of a BDNF-like small molecule compound as a potential therapeutic agent for KAND. I hope I don't get too technical in my talk, but please feel free to stop me at any point. I'll be happy to talk about anything specific that you would like me to talk about. So as you know, KIF1A is a motor protein that transports things inside the cell. This protein is especially important for nerve cells or neurons because neurons have long processes called axons. So this rounded, oops.
Red goes back, green.
I forgot. Sorry.
Oh, no, that's fine.
And this is the laser, right?
Yeah.
So this rounded structure here is the cell body. This is the nucleus. And this long process is the axon of this cell, axon of this neuron. And things need to get from the cell body to the tip of the axon, and from the tip of the axon back to the cell body for the neuron to function properly. I think you've seen this video already, but yeah.
I somehow pasted two on top of each other, so.
Thank you.
Oh, I'll give you back the cute sound. I just haven't listened to the sound, so I don't know what it's at. Don't blame me.
So this is the KIF1A molecule. It's transporting this gigantic vesicle filled. This is the cargo of the molecule.
Sorry. I'm just muting it. You can keep. Whatever.
Can I
it
go to
should
the
be.
next
Yeah,
slide?
You're good. Hold on. Well, I will make this work. Okay.
Okay. So one of the proteins or one of the cargoes that is carried by KIF1A is brain-derived neurotrophic factor or BDNF. And this is a factor that is really important for nervous system development, maturation, as well as function. Mutations in KIF1A lead to decreased trafficking of this cargo, decreased trafficking of BDNF, and we believe that lack of sufficient amounts of BDNF contributes towards KAND pathology. So our lab focuses on brain, and there are two different ways in which we look at the brain. First approach is in utero electroporation. In utero, in the uterus, electroporation is a process by which we get the DNA inside the cell by applying a small electric pulse. So what we do is we do a C-section surgery on an anesthetized pregnant rat. We expose its uterus, inject the developing fetuses with the DNA, and apply a small electric pulse to the brain. Then we reposition the uterus back inside the mom, allow it to survive for next 3 or 4 days, at the end of which we sacrifice the mom, take out the uterus or take out the fetuses, and analyze those fetuses, the brains of those fetuses. And the second way is, of course, the mutant mouse. As you know, we have a KIF1A mutant mouse. We are breeding this mouse, leg dragger mouse, and we analyze the brain of this mouse. So here, in this condition, we are inducing the symptoms of KAND, whereas this mouse already has the symptoms of KAND. It cannot walk properly. It has hind leg paralysis, and its brain is shrinking. It has brain atrophy. So let's begin with the first approach, in utero electroporation approach. And again, stop me at any point. We inject the DNA. The DNA is going to deplete KIF1A. So it's going to create conditions such that there is not enough KIF1A to do the function. And brains harvested after this procedure have fluorescence. So these green cells here are the cells which lack sufficient amounts of KIF1A. And on the adjacent panel, we have stained for a specific protein called TBR1. The name of the protein is not important, but this red band here spans the entire width of the cortex, whereas in normal conditions, under normal conditions where there is enough KIF1A present, this band is supposed to be restricted only to the superficial layers of the cortex. But instead, it is now spread throughout the cortex. However, when we treat this brain with BDNF, you can see that this band is restored back to its expected position, back to its normal position, to its superficial layers. Sure.
So, the BDNF is what the KIF1A carries
Correct.
in a normal KIF1A.
Correct.
And for our kids, they are missing BDNF.
Probably, yes.
So what you're finding in your study is that you put the BDNF back,
Correct.
and your fourth picture is showing how it should look after you put the BDNF back.
Absolutely. Correct. Thank you. So as you can see, BDNF is a potentially good therapeutic agent. It could be a good therapeutic agent, except the problem is if you inject BDNF, it does not cross the blood-brain barrier. So it is not able to reach the brain, or it is not able to reach the spinal cord. Also, if you swallow a pill of BDNF, it is digested by digestive enzymes. So again, it does not reach the brain or the spinal cord. So what we do instead, we found these BDNF-like small molecule compounds, which can be injected and they'll reach the brain or the spinal cord, or you can take a pill and again they'll reach the brain or the spinal cord. And interestingly, these compounds show the same effect as BDNF. As you can see, this in the condition where there is not sufficient BDNF due to lack of KIF1A, this red band is spread throughout the cortex. But when you treat this brain with these BDNF-like small molecule compounds, this red band is restored to its normal position where it normally should be. So we have this compound, and we have the mouse. So the next thing we tried was use these compounds, feed the mice with this compound, BDNF-like compound. But before we look at the results, we have to first look at how the brain of the mouse looks, how the mutant mouse brain looks. This is the, this is the normal mouse brain. This is a brain section, and these are the back outline. This is the outline. It's a little faint, but hopefully you can see it. And these at the center are the fluid-filled cavities, which are expanded in this mutant mouse brain. And these are expanded because the width of the cortex, so cortex is this structure here. The width of the cortex is slightly less compared to normal animal. And you can see this better in these sections here. These are just enlargements of, so this is the enlargement of a normal brain, and this is the enlargement of the mutant brain. And as you can see, this width of the cerebral cortex is slightly smaller, and because of which these cavities are expanded. Ventricles, these are called ventricles, these are expanded. There is another way to look at this, and we stain with a particular stain which stains neurons or nerve cells. And these black dots, again, this is a normal brain, and this is the mutant brain. And these black circles encircled by bright green color are individual neurons. And if you count neurons in the normal brain and in the mutant brain, you can see that this is the quantification indicating that there are fewer number of neurons in the mutant brain compared to the wild type or a normal brain. So now we treat the brain, we treat the mouse, we administer this BDNF-like compound in the drinking water to the mouse. And what we see is that these cavities, these fluid-filled cavities, are still a little larger compared to the control condition, but they're not as large as what they were before. We also looked at the neuronal number, and again, these tiny circles here are individual neurons. The neuronal number was restored also. So there are comparable numbers of neurons in the mutant as compared to the normal brain. So these are promising studies, but we need to, these are very preliminary results. So we need to conduct future studies to assess the role of this BDNF-like compound as in the treatment of KAND. I would like to thank my mentor, Dr. Richard Vallee, and all the members of the Vallee Lab. I would also like to thank Dr. Wendy Chung and Chung Lab. And I would like to thank all of you for giving me this opportunity to share my research findings with all of you. Thank you.